Bismillahirrahmanirrahim. We start paper 5. Now, the discussions on the paper 5, we have had a few papers which I've discussed. Now, I'm trying to do all the new ones which are for the year 2022 when the syllabus was changed. Now, as you can see here, the marks for this are 30 marks, but the main thing is the timing. It's 1 hour, 15 minutes. Now, another thing which you have to understand is paper 2 is on the same day, and that is for 1 hour and 15 minutes, but that is for 60 marks. And paper 2 are the ones who students who have not done the AS and will be taking that. Similarly, paper 5 is on the same day, and that is also for 1 hour 15 minutes. But look at the marks. The marks are only 30 marks. Now, why is this? Why have they given you only, this paper is for 30 marks. Why have they given you the same time for less marks? So paper 2, 1 hour 15 minutes, 60 marks, but paper 5, 1 hour 15 minutes, 30 marks, so that you can, really read the question, understand the question. It's more of a thinking paper. So you need to think a lot. You don't have to come out and say, oh, miss, I finished the paper half an hour earlier, and I would be very angry if you did that. Because that means you didn't even bother to read the question carefully and answer, because they're very difficult questions. And then you need to spend that whole time, 1 hour, 15 minutes on the paper, read the question, read it again, then think of the answer, then go on to the next part, read the whole question, and then go back to the question. So you need to read the question, understand the question, and then think of a befitting answer. Now, there are usually two questions, sometimes there are three as well, but there are usually two questions in most of the papers. Now, let's read the question together. Antibiotic resistance in bacteria is a global problem. Now, this is part of the AS that has caused scientists to research into antibacterial substances other than antibiotics. Honey has properties that make it a good antibacterial substance. For example, honey contains hydrogen peroxide, which is known to kill bacteria. So, honey contains hydrogen peroxide, H for honey, AH for hydrogen peroxide. The most effective honey tested so far for antibacterial activity is Manuka honey. So, there's a special type of honey which a special type of bees will make, which has antibacterial properties. It contains less hydrogen peroxide than many other types of honey, but it does contain an antibacterial compound. So besides the hydrogen peroxide, it contains an antibacterial compound, methyl glycol, and that the abbreviation for this is MgO, not OMG. Yeah, don't laugh now. OMG, but MgO, which is not found in other types of honey. So besides the hydrogen peroxide, it contains MgO, which actually kills bacteria. It says it has antibacterial activity. A student decided to investigate the effect of two antibacterial substances on the bacterium Bacillus subtilis, which respires aerobically. MgO in Manuka honey, an antibiotic solution used in cell cultures to prevent contamination. So the student decided to investigate the effect of two things. One was MgO and the other was an antibiotic solution used in cell cultures to prevent contamination. When we are sort of growing bacteria and all that, we need to culture them. We prevent, no, sorry, in cell cultures, not bacterial cultures. We don't want certain bacteria to grow, so we use an antibiotic solution. Now, I hope you all remember from the AS level what is an antibiotic. Like you all had augmentin or ciproxin or vibramycin, that's an antibiotic. The student wanted to find the lowest concentration of each antibacterial substance. Lowest concentration of MgO, lowest concentration of this, that would kill or inhibit the growth of B. subtilis. So there's one bacteria called B. subtilis, or we can say BS. Surprisingly, what does BS mean? Beautiful story. I'm sure you didn't think what I was thinking. So A part of the question, the student used a broth culture for the investigation. To make a broth culture, a small quantity of this bacteria B. subtilis, BS, is added to a clear nutrient solution. So there is a nutrient solution which contains glucose, amino acid, but it is a clear solution. A fresh newly made broth culture of B. subtilis is also clear. Now figure 1.1 is a diagram of a fresh broth culture of B. subtilis. There is a cotton wool bung and there is a fresh broth culture. And what is the broth culture? Is a small quantity of the bacteria and the nutrient solution. A small quantity of the bacteria and the nutrient solution. And it is clear, like we say water is clear. We don't say water is transparent, we say water is a clear solution, right? And then it says a sterile cotton wool bung was used in the top of the flask containing the fresh broth culture of B. subtilis. So we've got the BS here, the bacteria here, and we've got the nutrient thingy here, the nutrient solution, just like it's the food for it. I told you it's got glucose, amino acids, and ions and water in it. To protect the culture from contamination, explain why it is better to use a sterile cotton wool bung in a flask containing broth culture of B. subtilis rather than using a sterile rubber bung. So basically, it's common sense. Wool bung, so air can pass through it. If it was the other bung which would make it airtight, then no air would be able to come into it. So why it is better to use a sterile cotton wool bung? Sterile means that it's been boiled so that there are no bacteria present on the cotton wool. So basically, it is to allow oxygen. Because where did it tell you? Why will it need oxygen? Oxygen to enter for respiration. We can't have it in a vacuum. We can't have it in so basically the air will enter and the air contains oxygen because the bacteria respire aerobically. You can see this was given in the question that the Bacillus subtilis respires aerobically. So you actually read through it, but if you didn't read it carefully and you didn't register it, then you would not get this right. Because it says, why did we use a sterile cotton wool bung instead of a sterile rubber bung? The rubber bung would be airtight. So that was the answer to that. Now coming to part 2 of the question, before comparing the two antibacterial substances, the student carried out a trial experiment. The student transferred a sample of fresh broth culture to a culture tube and incubated the tube at 25 degrees for 24 hours. So 25 degrees Celsius, 24 hours, please remember this. Figure 1.2 summarizes the results of the trial experiment. 15 centimeter of the culture tube containing the broth culture incubated at 25 and it became a cloudy. This, remember, was clear in the beginning. And now it became turbid or cloudy. If you don't know the meaning of the word turbid, then they've explained it to you that meant cloudy broth culture. So after incubating it, now it has become cloudy. It is not clear. The student decided that turbidity of the broth culture is a measure of bacterial population growth. So more the growth, more the turbidity. More the growth, more does it become cloudy. Explain how this concept can be used in an investigation to measure the extent of bacterial growth. So how would you measure it? As the population or the growth of bacteria increases, the more turbid or the more cloudier it becomes. So somehow if we could measure the turbidity or the cloudiness by the human eye against a cross in the background, if we put a cross in the background and we looked at it, could we see the cross? Could we see the cross clearly? Could we see it dimly? So, or we could use a turbidity meter, or we could use a color chart. So now this is something which you had to really think and imagine. And I always say when you're doing a paper 5, think that you are doing it in the lab. So how could you measure turbidity? Well, there could be a turbidity meter. There could be, if you just put make a cross on a piece of paper and hold it behind this and see, then, okay, you can't see the cross at all. You can see the cross a bit, and then you could make a measure of it and say, okay, 1 to 10, the more the turbidity, it is this 1, or it is 2, it is 3, it is 4, like arbitrary units. So the student decided that turbidity of the broth is a measure of bacterial population growth. Explain how this concept can be used in an investigation to measure the extent. So as growth increases, the cloudier the culture broth becomes, or you can say more turbid it becomes, or you could use a turbidity meter, or even you could use a colorimeter. Even a colorimeter would be fine. Colorimeter measures the amount of light passing through a colored fluid. So less, when it's less turbid, more light will pass. When it's more turbid, less light will pass. Then coming to the B part of the question, the student decided to test the antibiotic solution before testing the Manuka honey. In addition to normal laboratory apparatus and materials, the student was provided with a fresh broth culture of B. subtilis, a clear antibiotic solution, nutrient solution to dilute the antibiotic solution, 15 centimeter cube of flat bottom glass culture tubes with sterile cotton wool bungs, a choice of graduated pipette to measure volumes accurately 0.2, 2.0, 10.0 and 25.0 cm cube. The student prepared dilutions of the antibiotic stock solution and added a volume of each to different culture tubes. Added a volume of fresh broth culture of B. subtilis to each culture tube. Incubated the culture tubes in an incubator. Allowed time for bacterial growth to occur and then checked each culture tube. Recorded and analyzed the results. Decided on the lowest concentration of antibiotic solution that appeared to kill or inhibit the growth of B. subtilis. So you first prepared the dilutions of the antibiotic stock solution, so you would have different dilutions, but the volume would remain the same. Then you added a volume, again a fixed volume, 2 cm cube or 3 cm cube of the broth culture to each culture tube. Then you incubated them, means you kept them in a water bath at a specific temperature, like as I told you in the beginning, 25 degrees Celsius for 24 hours. That is something which you picked up from the question. And then you allowed it for 24 hours, and then you checked it after the 24 hours. Then you recorded and analyzed the results. And then you decided on the lowest concentration of antibiotic solution. So, which would be appear to kill or inhibit. So, outline a control. Now, again, now when I say you must know what is a control, it's not that in this question you should know what is a control. You must understand what is a control, what is the concept of control. Basically, it is to compare. If I say this plant is producing oxygen, so then I keep it in a closed environment and it produces oxygen. I have some oxygen meter and checks that. But if without the plant also oxygen is being produced, then that means that plant is not producing the oxygen. So you replace the antibiotic with water, and if that even kills the solution, or you just put nutrient solution, so you were actually checking for that antibiotic, whether it kills the bacteria or not. So if you just put replace the antibiotic with distilled water, or you can say replace the antibiotic with the nutrient solution, and if that also kills the bacteria, then that means the antibiotic wasn't working, it was just something else which was working, which we didn't figure out. So basically, what is a control? In an enzyme practical, it's usually, you remove the enzyme or you boil the enzyme. But here, boiling the antibiotic is not going to kill the antibiotic. So you replace the antibiotic with distilled water or you replace with nutrient solution. So basically, you should know the concept of control. We teach it to you even in O levels. We keep on telling you about the control in photosynthesis chapter and all these different chapters. So you must know what is a control. So how would you word it? Replace the antibiotic with distilled water, or you can say replace it with the nutrient solution. Now coming to the C part of the question, in the next part of the investigation, the student used a stock solution of Manuka honey. The student remembered that hydrogen peroxide could be present but could not think of a way to break down the hydrogen peroxide to remove it from the solution. Describe how the student can improve the investigation by removing hydrogen peroxide from the Manuka honey solution and explain why this improvement makes the results more valid. Yeah, because you had besides the hydrogen peroxide, it had another antibacterial. So what you had to do is you had to understand is you can improve by adding catalase. What would catalase do? Now this is something we studied in AS level. If you added catalase to it, it breaks down hydrogen peroxide. So the catalase will break down hydrogen peroxide into water and oxygen. So the catalase enzyme would result in breaking down the hydrogen peroxide because we wanted to remove it from the solution. So you added catalase, or you could have even said add peroxidase, hydrogen peroxide, peroxidase. Why are you doing that? Because what will happen? You give an explanation because it says you can improve the investigation by removing hydrogen peroxide and explain why this improvement makes the results more valid. So read this explain. You have to give me some answer for the explain. Yes, this was for you can improve by adding. This is for the improve part. How you can improve. It said here how you can improve. And then you have to give me an explanation as well. So the can improve is adding catalase and then the explanation. What is the explanation for this? The explanation is the results, which is the killing of the bacteria, is only due to the honey or the MgO. So then we know that the bacteria is not being killed due to the hydrogen peroxide. So killing the bacteria is only due to the honey slash MgO. Or you could have said is the killing the bacteria is not due to the hydrogen peroxide. It must be the other thing which is killing it. D part of the question, the student was provided with a stock solution of Manuka honey containing an MgO concentration of 600 micrograms per cm cube. This was a clear solution labeled 100% honey. So 100% honey is 600 micrograms cm cube. The same apparatus and materials were available. Describe how the student could prepare a 10% solution of honey using the stock solution. Construct a table to show how the dilution is made for the 10% solution and the other concentrations that the student could use. Now this table is something like we do in paper 3 table. So it wasn't a very difficult table. Now you realize 10% solution of honey is made by diluting the 100% stock solution with nutrient solution in the ratio of 1:9. For example, you take 1 centimeter cube of the stock and you add 9 centimeter of the nutrient solution. So you would make this table and you'd write it. You could make it 10, 20, 30, 40, 50, or you could make it differently. You could make it 10, 30, 50, 70. But the the space between them. So if you're doing a 10, then you do the 10, 20, 30. If you do a 20, then you do 10, 30, 50, 70, and so on. So this is how you would have had to make the table. There were only 2 marks for this table, but if you were going to do serial dilution, that would have been really messing it up. That would have been difficult for you. So it's better if you did a simple dilution, and this is what you would do because the stock solution was 100%. So how many centimeter cube would you add of the stock solution? How much would you add water? Here we're not adding water, here we're adding nutrient solution to it. That was given in the question. If you look at the question that was given in the question, that you would be adding a nutrient solution. Describe how the student could prepare a 10% using the stock solution and how the dilution is made and the other concentration the students could use, and then the same apparatus and material were used. Now let's look at the E part of the question. It says state the independent variable and the dependent variable. Now another very important thing is you must know what is the independent and dependent variable. If you don't know it, just Google it. I always explain it to you is that the independent variable is which I have changed. So what did you change? What did you decide to investigate? You decided to investigate that the Manuka honey or the MgO, the honey concentration is going to affect the rate of inhibition of bacteria or is going to increase the more bacteria to be destroyed or killed. So independent variable, dependent variable is always the result. What are you measuring? Now, you please remember dependent variable is you're measuring what? The number of bacteria which are killed. But how are you measuring it? The question is, how are you measuring the result? You're measuring the result by checking the turbidity. Is it more turbid? Is it less turbid? So how, like for instance, you're checking the rate of photosynthesis, but then how are you checking it? You're measuring it by the number of oxygen bubbles produced per unit time. So though you are measuring the rate of photosynthesis, but you do not write that. In the dependent variable, you write the number of bubbles released or produced per unit time. So dependent variable is always the result, and it is always plotted on the y-axis. So this is my mnemonic for it is dry. For the independent variable, it is the first column. Independent variable is always on the x-axis. Now, when you've written this, you will get your 2 marks for that. So these are the 2 marks which nobody should be losing. The independent variable is what you have changed. What are you investigated? If you look at 5 or 6 different paper 5s, you can figure that out in the question. It's a very easy thing which you can figure out. Independent variable. Dependent variable is the result, but how are you measuring the result? So how are you measuring the result will be the dependent variable. Then coming to the F part of the question, predict the results. The student would explain the prediction. Now always remember when you read a question, You must look at what is asked. Explain the reasoning behind the prediction. So first give the prediction and then give the explanation. So I've given it to you separately. Lower the concentration, the more turbid. Why? Because lesser the concentration of the Manuka honey, less of the bacteria. So bacteria will not be killed, so it will be more turbid. Or you can say higher the concentration will kill the bacteria, so it will be clear. It will be a clear solution because the turbidity is because of more the bacteria can grow. If the bacteria are destroyed, then it becomes less turbid or it becomes clear. Explanation is MgO inhibits the growth of bacteria. So lower the concentration, less the ability to grow to kill the bacteria. So the more the bacteria, more the turbid it becomes. Then coming to the G part of the question, describe how the student could determine the lowest concentration of Manuka honey solution that would kill or inhibit the growth of bacteria. Let's just keep it to kill the bacteria. Do not repeat any details given in D of how to prepare the different concentrations of Manuka honey, and some of you will do that. So I do not like those students very much who do that, who do not read the question and make the same mistake. And then do not give details of using aseptic techniques to prevent contamination of the student, the environment, or other people. So you do not talk about these two things. Your method should be set out in a logical way and be detailed enough to let another person follow it. Now, if you look at this, this is only for, there are a lot of lines available to you, but this is only for 5 marks. So everybody should get those 5 marks. And most of the students did get those 5 marks because you've learned all this technique very well. You all have very good teachers. I often see you all blaming your teachers, but I think all the teachers take their best and do their best. So it's not very nice to be talking about teachers behind their backs because you're actually backbiting. If you have any problems with any teacher, you should have the guts to go and talk to them yourself. So it's not very nice. This is our society. We've got this bad habit of talking behind people's back, and this should be avoided at all costs. So coming to this question on how would you do this experiment? So same volume of the culture that you used and same volume of the Manuka honey. Then mix the honey and the culture. Then how would you measure the turbidity? You use a turbidity meter or you put a cross and you look at it. Then you make a record of which concentration the broth is clear or is the clearest. Then how would you maintain the temperature? Anything between 10 and 50 degrees Celsius. So say 20 or 30 degrees or 40 degrees. Then you leave it for how many hours? Anything between 12 and 48 hours was the correct answer. You could have said 20 hours, you could have said 25 hours, or you could have said 24 hours. Then because these are all enzymes working, so use a buffer to maintain the pH between 4 and 10. Then repeat at least 3 times to find the mean. This is something which you can write nearly in every planning and design experiment. And then you name a hazard and the risk and precaution. So hazard, risk, and precaution. The live cultures, the honey and the broth were all irritants and they could cause an allergy. So it's better if you wear gloves. So the hazard, risk, and precaution would be 1 mark. So there were 10 mark scheme points. And you could have given me any 5 of those and you would have got your 5 out of 5. Got this all written down for you now. Same volume of culture used, same volume of Manuka honey used, mix honey and culture. Now, this is something which you will not write. Why will you not write? Because you're not concentrating. As I said, you think of it, you're doing it in the lab. So, okay, you take 10 cm cube of the culture, you take 10 cm cube of the honey. So, I've always said when you're taking a liquid, always tell me the volume. So the same volume will be used and you mix the honey and the culture. Now this was a point which is very important that students will have missed out, not realizing that unless you think you're actually doing it in the lab and you're actually doing this experiment, then you can only think of this point. And then you measure the method. How do you measure the turbidity? Either you use a turbidity meter or you put a cross behind it or you sort of measure some other calorimeter. Now, the other points are any two of these. So these 4 points were absolutely essential points. And you had to give me two of these or one of these or three of these at least, yes, because there are any two from the other ones. Now, from the other ones is maintain temperature using a water bath at 30 degrees. You use the same time left in the incubator, leave it for 20 hours. Anything between 12 and 48 hours was correct. Use a buffer solution to maintain pH at 7. Repeat 3 times and find the mean. So repeat 3 times and find the mean is one that you must give in every planning and designing. Then the hazard and the risk and the precaution. So live cultures can cause infection or an allergy. So you wear gloves or goggles or mask or ppE. So any two from these points which I've given you at the end, any two from these points. Now, coming to part H, MRSA, methicillin-resistant Staphylococcus aureus is an example of antibiotic resistance in bacteria. There is evidence that medical-grade Manuka honey is effective in treating wounds infected with MRSA. This honey has been sterilized by gamma radiation and filtered to remove contaminants. A study was carried out to see another type of honey, Germania honey, is as effective as Manuka honey in killing bacteria removed from wounds of 50 people with MRSA. 5 different concentrations of each type of honey were compared. The concentrations were numbered 1 to 5, with 1 being the highest concentration and 5 the lowest concentration. All the concentrations, uh, all the concentration where there was no visible growth in a broth culture of S. aureus, the researchers transferred samples onto nutrient agar plates containing no antibacterial substances. Incubation of these plates confirmed that there was no bacterial growth. The results were analyzed using the chi-square test. Table 1.1 shows the result of the study and the statistical analysis using the x-square test. So we've got all this result here. Table 1.1, you can see the values. Now state a null hypothesis. Now this is something which we've studied many times before. State a null hypothesis. Null hypothesis means you negate it. So there is no difference in the effect of Manuka honey and Germania honey. So you just have to say there is no difference between whatever and whatever. So that is called a null hypothesis. You must read up on it and you must understand it. And you do a number of 10 questions on null hypothesis, you figure it out what it means. There is no difference in the effect of Manuka honey and Germania honey. Now coming to the last part of the question, part 2, table 1.2 shows some critical values for x square at different probabilities. Now we always look at 0.05 probability, that's the rule in biology. And because there were two, so n minus 1 you would be looking at 1 degree of freedom, and that is 3.841. Now when you look at 3.841, it says use table 1.2, which is this one, to decide whether the x square values for concentration 2, 3, and 4 in table 1.1 are significant or not significant. Write your decision in the final column of table 1.1. Write yes if the value is significant, write no if the value is not significant. Now how do we read this? Now you have to understand how we read this. You see, if the value is more than the critical value, then you reject the null hypothesis. That means it is significant. If the value is less, then you accept the null hypothesis, which means it is not significant. So here you have to write, because this is all more than 3.8. This is more than 3.8. So this is yes, yes. And this is less than 3.8. So it is no. This was also more than 3.8. So it is. So you, when the value is more than the critical value, and how do you calculate the critical value? N minus 1. And you look at the probability of 0.05%. So 0.05 degree of freedom, you have to decide it is N minus 1. So there were two sets of figures, and so 2 minus 1 is 1, and you look at 0.05, 3.841. So you look at it, if it is more than 3.8, then it is significant. If it is less than 3.8, it is not significant. So that is why we wrote yes, 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 and no, and no. Now, last part. The study compared the effectiveness of the two honey varieties in treating wounds infected with MRSA. State the conclusions that can be made from the results and statistical analysis of the study. Now you can understand that the study compared the effectiveness. So what do you see? We see Manuka honey kills more bacteria than Germania, because you can see it from the table. And the main thing is you have to look at the statistical analysis. So the first three are significantly different, which you can see here. Yes, yes, yes. These are significantly different. So these had more concentration of Manuka honey than Germania. So that is why you have to refer to the statistical analysis to get your 2 marks out of 2. So this is the end of this paper, and we'll continue with the question 2 in the next video, and wish you all the best, and thank you very much.